Thank you, Wendy. All right, well, this is quite exciting. Thanks to everybody, both in person and online, for joining us today. For those folks who are in person, we have some sweet treats over on the table. So hopefully you grabbed your lunch and you can grab a little dessert. We are recording this. We have several folks online, which is great. Welcome to everyone online. So let's get started. So my name is Emily Elo Phadrash. I'm the lead of the National Microbiome Data Collaborative. I'm really excited for this session today. We have a lineup of 3 great speakers, and then we also have some very exciting updates that the NMDC has been working on over the past year, and especially over the past couple months that we would like to share. So looking at the crowd and then maybe the folks online, maybe the slide was not completely necessary, but I just wanted to let everyone know what is the NMDC. So we are a program focused on how we can support researchers to create, use, and reuse microbiome data. We've been in operation for almost 5 years now. We started as a 3-year pilot program, and for the past year and a half have been in what we call production phase for the software and data services that we offer. In the corner here, this is an overview of the 3 products that the NMDC has: the submission portal, NMDC Edge, and the data portal. And really the goals of what NMDC is trying to accomplish is using data standards, infrastructure, and community building to advance microbiome research. And so here's a picture of our team from our latest retreat that we had back in October in Monterey. Over the past year, we've been very active in building out our infrastructure platforms. You will hopefully have met Alicia and Julia, who had posters both yesterday and today at the same time. We're really excited about the amount of data that we've been able to pull into the data portal and thinking about how we can support both DOE researchers and researchers outside of the DOE for doing continental scale research. So we have several projects and programs and collaborations that we've worked with very closely for enabling microbiome data to be made more broadly available. We are also really excited this past year to onboard 24 really phenomenal early career researchers as part of our ambassador program. We've effectively doubled the ambassador program, so this is really exciting, and we're really looking forward to these really amazing researchers working with our team over this next year across a whole range of really diverse microbiome studies. We released back in January our annual report, so I encourage you to look online. We also have some hard copies in person to read more about the work that we've been doing. And so today, for this town hall, we have a lineup of 3 really amazing talks. Josué will be talking about his work as an ambassador this past year using the NMDC Edge platform. Buck, who has been a champion with our program for the past couple of years and is now an ambassador for this 2024 cohort, will be talking about bacterial fungal interactions. And then Neslihan, who's a wonderful collaborator, will be talking about her wonderful work in Arctic soils and permafrost environments. So I'm really excited to learn about the science and how they're working with our team to really advance the microbial sciences. And then we have some exciting news. We will be previewing the NMDC Field Notes app. We've been promoting this as Sample Different. So without, I guess, getting too much into the details, I think this is going to be a really exciting way that field researchers, lab researchers can really use standards and collect metadata in a standardized way that will advance the metadata and data lifecycle for microbial studies. So I think this is a really exciting effort. I'm super proud of the team for bringing this all together. And with that, I hope that I haven't gone too far over time. But we will go to Josué for our first talk.
Which is the thing that advances?
So just the big green, and then if that's back. Yep.
I see. Cool. Pointer. Okay. Hi, everyone. I'm Josué Rodríguez. Thank you all for coming. And today I kind of wanted to talk to you about what I had done as an NMDC ambassador and how I leveraged my undergrad institution, which I did my bachelor's degree in Puerto Rico, into NMDC Edge and using some of these tools that we have developed. And so my title is Charting the Unknown, Leveraging the NMDC Edge to Illuminate Puerto Rico's Ecological Diversity. So a little bit about me. I'm a postdoc at the Pacific Northwest National Lab right now. I have a PhD in ecology from the Colorado State University and a bachelor's degree in industrial microbiology from the Universidad de Puerto Rico, Mayagüez campus. And my research focuses on understanding soil microbial, viral, fungal interactions, how they respond to stress, and how these communities evolve under different conditions in soil systems. And so as part of the 2023 NMDC Ambassador Cohort, you're required to do these 2 workshops. They can be one big one, one small one, and usually we do workshops at conferences like this one or conferences like ISME or the JGI User PI meeting. I did one at the JGI PI meeting with Thomas Pitod, but for my second one, I wanted to do something that was a little bit different, and I wanted to leverage, like I said, my experiences in my undergrad institution and my connections to see if we could do something over there. And so a little bit of more background about me. I started my scientific journey back in 2015 at the Microbial and Biotechnology Laboratory in Puerto Rico in the laboratory of Dr. Carlos Rios Velázquez. I was an undergrad student, unpaid, fully volunteer labor, had no idea what metagenomes were, and I mostly focused on bioprospecting and identifying genes in bacteria that could help us maybe promote some biotechnological discovery that might be useful in the industry. But I knew from my time there that my professor was always very interested in metagenomes and that there was a handful of them that existed within the lab, even though I never personally worked with them. And so for my second kind of workshop, I wanted to talk to him because we're still friends, we have a good relationship, and collaborate with the University of Puerto Rico to see if we could do an NMDC workshop there. And I told NMDC that I wanted to do that, and they were like, this sounds awesome. How many people do you think there are gonna be? And I said, maybe 0, maybe 40. I don't really know. We have no gauge for this, honestly. But they were like, you know what, this is still a great idea, you should pursue it. And I did. And so I met with Carlos Rios, I showed him all of the materials that NMDC had, and I said, hey, this is what I'm planning to present. And he was like, I'm very excited, this is awesome, but this is all in English. And I was like, this is all in English. I need to translate all of this into Spanish. And so I said, give me a week. And so I took all of the NMDC slides, all of the NMDC material, and I translated it all into Spanish. And then I said, okay, we're ready. I gave him my preemptive presentation, and he was like, this is great, but nobody's gonna understand what you're saying. You need to teach. You need to teach what metagenome is. You need to teach what reads are. You need to teach all of these principles of things that we kind of take for granted in order for them to understand. And I said, oh my God, okay, how are we gonna do this? And he's like, well, I give a bacterial genomics class. I will give you a 2-hour lecture slot. And I was like, okay, thanks, I guess. But we did it. And so I gave this 2-hour lecture on basic metagenome multi-omic concepts. I used this puzzle analogy to explain how assemblies work and how bindings work. And with this, he was like, okay, we're ready. Let's do the workshop. And so almost a year to this day, April 13, 2023, I gave my NMDC workflow on NMDC Edge. And a few people came, I think it was like 20 people. And as part of that, I said, hey, bring your own data if you have it. And 2 of these students, so Edwin Rivera Lopez and Gabriela Melendez Martinez, they were like, we have a data set. We can put through the NMDC Edge as we go. And I was like, great, what is it? And they were like, it's a sea slug. I was like, what? Why? And I was like, yeah, it's a sea slug. It's Holoturia glaberrima. I was like, what? Why? So I was like, sure, bring it. And then I was like, in my head, I was like, why do they have sea slug metagenomes? Well, it turns out that this is a biological model for regeneration studies. And to that I said, what? Why? I'm so confused. What is happening with these sea slugs that's so interesting in this way? Well, it turns out that these sea slugs have a really interesting intestinal system. They have these different compartments within their intestine that all have unique microbiome communities. And this is a paper that I found from another group from the Universidad de Puerto Rico, Rio Piedras, where they cataloged kind of the microbiome community and showed that they were distinct organisms in each of these different compartments. Now, when this organism is stressed out or when it ingests a toxin, its escape mechanism is to eviscerate its digestive system and expunge it into the environment, which is insane. But what's even more insane is that after this happens, once it detects that the stressor is gone, it can pull it right back in and reform all of those different digestive compartments down to the microbe. And that's why it's an interesting for regeneration studies. But they had these data sets where they have metagenomes of each one of those different segments before and after the evisceration. And so they were understanding how the community looks similar or how it looks different. But they didn't have the ability to assemble these quickly in-house. MGRAST had just fallen and they were like, I have no idea what we can do with this data set. But with NMDC Edge, we were able to run it through that pipeline, and we were able to get what I think is the first publication that uses the NMDC Edge as their sole pipeline for annotation and assembly. And if you watch The Office, this is a win-win-win by Michael Gary Scott here. We win as NMDC because we have a publication. We've shown that this thing works, and we show that we can engage people from undergraduate institutions that are minority serving that aren't even our primary user base, but maybe they should be. But they win because they get a manuscript, and this is a teaching laboratory. This is undergraduate students that have a paper now that can go on and learn some more things based off of what they're doing. And so it's just really useful. And so, like I said, it's a teaching laboratory. Like, I remember being here. I came from here. And these are the capabilities that can get people ahead and be able to help us get to where we need to get. And so you have all of these students that can get more involved in the studies that they collect the sequencing for, but you can get them to actually play with that data and understand that data a little more. And now this has kind of evolved into more work for me, because Carlos Rios was like, well, we have all these other data sets. And I was like, well, let's start writing them up, man. Like, let's do this. Like, we can do this. And so they have these microbial mats from the Observatorio Microbiano de Salinas in Cabo Rojo. It's like an observatory that they built. They collect these metagenomes to try to use these as analogs for early life. And this is awesome because I remember when I was an undergrad, I started the Association of Exobiology, and nobody came ever. But it's interesting that now people seem to be getting on and carrying up some of these different things that I was interested in when I was there as a student. And so we're working on this manuscript right now. We're working on getting this published with these metagenomes, again, through the NMDC Edge. And this is really cool for the future. And so I didn't know this, but Puerto Rico has the third largest underground cave system in the world. And Carlos Rios and a few other scientists are working together to try to taxonomically and functionally catalog some of these cave systems. And so they have 16S data, they have ITS data, they haven't had funding for metagenomes yet. But as we know, this is the GSP. Once you show that you have the capability for something, getting funding for that is a little bit easier. And so now we're in a process where maybe we can start writing up some proposals to get the sequencing information that we need so that they can continue to do the science that they're already doing, but just more efficiently and in a better way. And so with that, it's 2 more samples in this NMDC data portal from Puerto Rico, from Puerto Rican scientists. And I think that that is very important to remember that, yes, as NMDC, it's very important that we continue to push the forefront of the field, but that our user base exists in all stages of where we are, and that we need to meet our users where they are. And this is an example that these users want to engage with us. Like, even the review comments from that manuscript that we submitted was incredibly helpful and really awesome. And they were all very excited. We've never heard of the NMDC. I saw that you can make your workflow public. Can you make it public so that I can look at it and we can run some stuff on our end? So there is a user base out there that maybe we don't access in some of these meetings, but will engage with us and will help us with our goals and their goals. And with that, I wanted to put Carlos Rios' picture up there. He said, please just give them my email, give them my Instagram, because they don't do Twitter, apparently. So if anyone wants to talk to him, he's very friendly. He's a very great guy. or email me and I can put you in contact with him. But thank you very much. And if you have any questions, I'd be happy to answer them. Yeah. So the NMDC Edge is more standardized than KBIS in the sense that you don't need to kind of develop your own narrative and figure out what you need to do. You give it the reads and it uses a very standardized platform to process those reads, and it gives you an output based off of that data. And so for a user that is not super familiar with K-based narratives and understanding what exactly might need to be done, I think that the NMDC Edge is very useful in those use cases where I just have a FASTA file or a FASTQ file of sequencing. I can trim it, process it, and have an output ready, and then upload that output into the NMDC data portal for future access of other scientists without having to worry too much about how I get there, because I know that it has been vetted and standardized in that sense. Yes.
So that was a great talk. Your data sets are so fascinating and unique. Did that kind of cause any issues for you when you're trying to kind of provide? Yeah. So your data sets are really fascinating and very unique. So I'm wondering, did that cause challenges when you're trying to submit metadata? Like you were trying to look for, okay, where's the right term to describe the eviscerated gut of a sea cucumber?
I was thinking about that this morning, and I was thinking, hey, I need to email Emily, because we're now at the stage where it's finally published. This morning, we resubmitted the revisions, so it's done. And now I'm like, okay, we're ready to release it. And then I was like, I don't think sea slug was a term in the metadata table. And so it's going to be interesting to figure out how we can leverage some of these weird ecosystems, like you say, into the metadata table. And so now that we have this concrete example, what do we need to do to make it work and make it a little more accessible for some of these weird use cases?
You could just update it, right? You could just be like, okay, this is all weird and different.
Yeah, what do you need? And then, I don't know, this. Is it a male or a female? I don't know. They know, probably, but I don't know. Slug compartments is a term that we need, and never thought you'd see that, huh?
Your video is really great.
They sent it to me, and I was like, I'm showing that. That's gross. Okay, thank you very much.
Great. Thanks, that was awesome. So I do recognize that there have been some registration challenges with people trying to get online. We're trying to fix that as fast as we can, but fortunately we are recording this, so we'll be posting it later. So next up we have Buck. So
Hello, everybody. My name is Buck Hanson. I am a scientist at Los Alamos National Lab, and my main project there is to investigate bacterial and fungal interactions and how they can be utilized to improve plant productivity and plant ecosystem services, particularly with carbon sequestration. Forward? Yes. No. Back. What did I do? Pushed this red one.
oh, that's weird.
Okay, big green
So
one.
just the big one.
The biggest one. So for those who may not study plants and rhizospheres, there are many beneficial interactions that plants can create or induce or recruit beneficial microorganisms to their plant roots so that they can be resilient to abiotic stresses, enhance nutrient acquisition from soils. They can excrete exudates that then can cultivate a specific microbiome that's associated with their roots that can ultimately increase plant productivity, resistance to pathogens or immune system priming. But for our purposes, in terms of studying this system, the plant microbe soil interaction is for understanding how we can leverage these interactions to potentially increase soil carbon sequestration. So for this, we have been studying a model system, the oxalate carbonate pathway, that involves bacteria and fungi that are associated with plant roots that excrete oxalate as a root exudate. So these plants, some plants are able to do this, some plants do not. So one of our key questions is, can we identify the components of the oxalate carbonate pathway that we can then leverage in the future and try to promote this type of plant-derived carbon into stable carbonate pools. So oxalotrophic bacteria and fungi can stabilize plant-derived carbon by releasing CO2 that then gets complexed into calcium carbonate and increases soil fertility by affecting soil pH. And there's also evidence that this contributes to a long-term carbon sequestration pools as oxalates. This is this one. So plants releasing calcium oxalate can be degraded by oxalotrophic organisms that then are contributing to this stable carbon pool. So understanding the players in this, let's say, carbon flow pathway, and how can we further promote this and understand it. Green button. So some of our big questions at this point are pretty basic for understanding bacterial interactions and soil microorganisms with plant roots and this pathway. So one of the things we know that the bacterial pathway for oxalotrophy is understood. However, we do not know what the fungal pathway for oxalotrophy is. And so do they compete for oxalate by using the same pathway or different? And how does this affect carbon flow? One of the things we also want to know is, can we track carbon from the atmosphere through a plant and into soil microbial populations as a way to track this carbon flow into these stable isotope or stable pools? So in an EMSL exploratory research proposal that was accepted in 2022, we took some model organisms that we know are oxalotrophic, so a bacteria and a fungus, and we were able to just grow them on oxalate as a pure carbon source, and we were able to show or find out that the bacteria, the bacteria produces about 40% of the original derived carbon from oxalate into CO2 and the rest into biomass. However, we saw very little fungal derived CO2, which was very interesting. We obviously saw growth of this fungi. However, when we looked at the metabolome of this using NMR, we saw that the fungi preferentially excreted formate into this rather than into the form of CO2. So this led us to the question of whether there is this differential pathway for oxalate carbon flow through bacterial and fungal populations that may have an impact on the overall sequestration of this oxalate carbon into carbonate pools. So if you're able to promote one microbial pathway, the fungal or the bacterial, can we then enhance flux of this carbon into carbonate pools rather than to a labile compound such as formate? In a second proposal that was funded or supported by EMSL as a research, large-scale research proposal, we wanted to take this a step further and investigate this in a model plant, Brachypodium, trying to understand the spatial patterns of oxalate as it's being excreted across roots, and then try to correlate the spatial localization of microbial populations with plant physiology, trying to understand if there is an association with potential oxalate hotspots and the microbial communities that are associated there. And then to finally funnel this up with some isotope labeling experiments to quantify carbon flow into these pools. So we did some pretty basic experiments up front where we grew Brachypodium in our modified bioreactor system so that we could do both sterile and non-sterile conditions. And you can see one of our team members, Leah Johnson, and I out here collecting soil samples from this arid grassland, which is one of our model systems, potentially future climate change type scenario. And we know that there are oxalotrophic organisms here, both fungi and bacteria, so we want to know more about how these might fit into this system. One of the interesting things from this work is that from these roots, we wanted to zero in on the spatial orientation of these microbial and the plant-associated interactions. So we did laser microdissectioning of these plant roots, and we were doing position-specific analyses looking at metabolites derived from the plant as well as the microbial community composition. So we can spatially correlate the plant physiology, microbial community composition, as well as the metabolomics that are happening that are associated with those spatial connections. And the final thing, just a few weeks ago, Leah and I were in PNNL, or EMSL, where they had done a 80 plant growth experiment using 13CO2, and we were looking at how we sent them New Mexico soils, and they grew these plants in their chambers for about 4 weeks. And then you can see us harvesting these samples, looking at the spatial orientation of potentially oxalate hotspots in these plant roots. And since these were 13CO2, we preserved samples here for our nanosims analysis. And then once we do some genetic, probably metabolomic, metagenomics and amplicon sequencing, designing some fluorescent probes. I keep hitting this wrong button. Designing some fluorescent probes that can associate specific taxa with those spatially quantified 13C coming from the plant. These are just idealized images at this point, but that's hopefully what we see in the future. So at this point, one of the big things we want to know is like, how do bacteria grow on oxalates? So looking at genes and metabolites from different pure cultures. Do root oxalate excreted from roots, does that shape the rhizosphere community structure? And how can we potentially understand these components so that we can be leveraging aspects of this to potentially improve carbon sequestration in desired plants that are associated with an oxalate carbonate pathway? I just want to say a few things about my engagement with the NMDC. I'm really excited about submitting these EMSL data sets to the NMDC portal as soon as they are coming in, and they will be the first data sets that are associated with this long-term ecological research site in the Servilleta, which is in New Mexico. It's the images I showed you where we were collecting our soil samples from. So I'm really looking forward to that and contributing data from this really important ecological research site. Additionally, as Emily had noted, I have been a champion for about 2 years, and I've been involved with user testing, testability, usability of the different products that NMDC has been developing. And as well as we recently had a kickoff for a writing team subgroup across the champion sub-cohort, where we are looking at the data that's already currently in the NMDC portal and how can we look at ways to reanalyze that data in looking across multi-omics or different sample sites, et cetera. And also we've been able to, last year we were participated as a panel at SACNES in Portland, where we discussed data management, tips and tricks, and all of our personal experiences with strategies and data management, and had a great discussion with 70 or 60 people who came to the panel. So I really appreciate your time. Thank you very much. And just a thanks to all of our team. I think we're running a bit late on time, so do you have a question real quick?
Are there any known pathway in the fungi that can convert the oxalate to formate, if you know any?
So, not that I'm not a fungal genetic transcriptomic gene expression expert, but as part of the study, we did have transcriptomic data sets that have been prepared, and we just the last 2 weeks received the genome for this fungal strain so that we can start looking into that, coordinating the transcriptomics with the metabolite data. Thank you.
All right, great. Thanks so much, Buck. And our last speaker today will be Neslihan. So the big green button back if you need, and then pointer.
Okay, thank you very much. Let's see. Okay. Hello, everyone. My name is Neslihan Taş. I'm a staff scientist in Berkeley lab, and most of my research is in Arctic soils and permafrost. Thank you very much for the invitation. It's a pleasure to talk to the community. We are going to start using NMDC real soon, but I just really want to give everyone a little bit of a flavor of the things that we've been doing so far, and just why we're excited to be part of this community. So permafrost is a bit of a peculiar type of soil. It's permanently frozen to be called permafrost. It covers a large landscape in the northern hemisphere, and it's rapidly changing. It's one of the most speedily warming section of the Earth, and it has a lot of carbon currently locked up in the soils from many, many millions of years of geological time. So some people call it like Earth's freezer. It really affects the whole climate in a sense. But the soil temperatures in there is on the rise, like many other places on Earth. So the permafrost, the frozen part, also gets warm in a parallel to the surface soils. So this Earth's freezer idea is more of a fridge at the moment, which is a very dangerous place to be. So what happens is that when this frozen soil thaws, it changes the landscape, not just the top, but the bottom, and all together, creating a very hydrologically complex environment to study. So most of you, are familiar with the, just the emissions that comes from any soil and how those whole that loop could feed back into. This is actually much more soil carbon than we currently know about. And one fine example is that how these environments could be very different from the others is that just this representation of methane bursts that you can burn them up. It just really functions in a completely different way than an just more temperate ecosystem. So what we have is a massive, massive carbon stock currently locked up, but thawing. We know better now, but especially when my research started, we were really not well informed about it. Even the risks of it, we're still trying to assess it well enough. So one interesting papers came out still a while back is that when you look at how current emissions coming from these systems and how climate change could change it is really very rapid. We were expecting to have actually a lot of input from these systems real soon. And the permafrost environments are really highly heterogeneous, and events in each and every scale actually has a huge impact. So all of these landscapes that you see in these beautiful pictures have permafrost under them. So one doesn't really look like the other one. And when you look at how the soils look like, there's also differently connected and disconnected from each other, where you have the active layer that goes freeze and thaw cycles yearly, and then there's this permafrost that's locked up, completely hydrologically separated from its top soil partners. And when you look at these soils in a granular level, it's all kind of different microbial habitats and different connectivity, just really adds up to the whole system. And we also have a very limited information about how life is when it comes to really in frozen conditions. We know that the microbes live in a state that it is still alive, not currently dead, up to -40°C, and they can grow up to -20°C in a very, very slow way. But once that state changes, when frozen becomes thaw, how they lose their water, how they interact with each other in the new hydrological system, and how that just brings it together to give a response to the atmosphere is a very, very hard problem to solve. So there's a lot of gap filling when it comes to the microbial part of these systems. We know that the permafrost systems are quite different than the topsoils, and when they thaw, the change is huge. But the end result of it is very difficult to predict, because at least currently we know that almost any biochemical reaction that we know of could happen in soil does happen in permafrost soils when it thaws. So the easiest thing that, just to figure out is if it gets wet, probably it's going to take a lot of input as methane. So I'll talk to you about a little bit on that. And then what happens to the remainder of the landscapes is an active area of research. But permafrost thaw response is very, very hard to predict, even with the help of microbiology. Most permafrost is assumed to be anaerobic and to remain anaerobic when they are thawed because of all the water becoming available. But there are places on Earth that the permafrost is actually fully populated, like in this location in Svalbard, by aerobic microorganisms. So when we analyzed this whole permafrost core going down from the topsoil to the bottom, we saw the trend that, oh, it's different than the topsoils, but we only just keep hitting more aerobic metabolism, which was really strange. And when we tested, oh, can they actually, are we seeing really just some kind of a really abundant population? No, they were just really able to do really very strongly aerobic carbon dioxide production. Even under anaerobic conditions, we were not really getting much production or methane in these systems. And anaerobic conditions also do not consistently yield methanogenesis. So this is a long-term research area in Utqiagvik, Alaska that I've been working over a decade now, that when you look at really very wet areas in this landscape, what you would expect to have some methanogenesis, but what we instead see a very strong fermentation that just really grabs that carbon and just emits that carbon dioxide. So you can have as much as carbon dioxide coming from the topsoils to the permafrost, but much more methane coming from the topsoils, where not really much coming from the permafrost. So this is really turns the system to a very big carbon dioxide emitter, which is different than what we would expect it if we didn't know the microbes that are living in there. So basically, in systems like this, we really need to start changing our way of thinking a little bit, relying a little bit more on the microbes to guide us, rethink our hypothesis. So this is a good example for me to just think about anaerobic doesn't necessarily mean methane production. It could also mean a lot of fermentation, iron reduction and sulfate reduction, and maybe a little bit methane. It really has a lot of consequences on how we approach to the, just the modeling. So, and I would like to also add that in this particular location, we didn't have any denitrification, which was really baffling. For a long time, we look at the reasons for it, but there are places that comes into play as well. So in thaw, sometimes anaerobic places become really even stronger emitters of carbon dioxide. So what helped is that we started to look for, just a larger perspective on this. Most of the research, as I presented to you, is one location, just trying to understand it very well. But what we try to do is to have a bit of a more pan-Arctic analysis of the permafrost with aid of collaborators. We looked at different locations and, just analyzed samples coming from 25 different Arctic locations. This effort was mainly led by Rachel McElprang and Mark Waldrop, but we were contributors in there, and there are some papers coming up from our side as well, that we looked at how the distribution of microbes were across the Arctic was, and what we found out that there was a nice interplay with the actinobacteria and proteobacteria, depending on where you are. What was interesting in this research that came out is that, just through the analysis of the genes in there, we could see how the distributions of microbes and important, metabolic processes could be found in the landscape, which was very interesting to me, that most of the soil parameters that we measure and hold very dear to us, like ice content, organic matter content, or nitrogen, did not consistently correlate with the microbial populations. What we found out that there was a very strong pH effect in the soils that most of the primary functions were correlated with. And there were so many things that we could explain and so many things that we could not. There was really a huge paleo-environmental effect in there that we didn't really, just couldn't really explain, but we would not have seen if we were not doing this big meta-analysis. So, just to, challenge the, just the thoughts on it a little bit. In most of the places, there is a chance that all of these populations and, just interactions with the environment probably is going to release more carbon dioxide than methane, and maybe in some places, methane production is going to overcome the carbon dioxide. But, just if something that we learned is that there's not one typical permafrost microbe that we can learn and, just assume throughout the landscape. that doesn't really matter if it's pan-Arctic or it doesn't matter if it's an site scale. So, and the predictive links between what microbes can do, what they do, and what we can predict is not really very strong at the moment, but it can be strong when you look at in a larger perspective in a systematic way. That's why we are also interested to contribute to the NMDC, to put our data into their systems. This is the data that's available as soil in the system. So, just Alaska is a really a large place, and now currently the representation is a bit small in there. So we hope to help to improve that a little bit, add our data in it. But, just more with the different data and metadata and measurements, we look at these systems, more we learn, and, just better we understand why things that are happening the way that they do. With that, I would like to, first of all, acknowledge the fact that most of this research is done in the land of North Slope communities of Inupiaq. We appreciate their help and support and access that they grant to us to do this research. Without their support, none of this could be happening. Most of the research that I showed you in here is a product of a next generation ecosystem experiment in the Arctic, which we got approved today, this morning. So we are very happy with our collaborators in Oak Ridge, Los Alamos, and University of Fairbanks in Alaska. And just Arctic research has to be a very, very collaborative research. Just access, sampling, just knowledge is really very hard to get. So I really appreciate all the work my lab does, but also my collaborators, the highlighted ones that I, just use their data today to, just give the story to you. And of course, thanks to the DOE and other funding resources that kept supporting us for all this times. And we're really excited to be part of NMDC. Oh, I don't know if I have time for questions. Okay.
Super interesting talk, thank you. I have zero experience working in Arctic soil, so this may sound like a dumb question. But I have done a little bit of research around the birch effect, right? So this idea that you've got really dry soil and you add water and you see that burst of CO2. Do you see something similar in Arctic soils, right? If they're frozen and they're not doing a ton and then suddenly they're thawed and now all of this water is available and that soil is actually wet from water, not just ice, right? Do you still see the same birch, and is there that same response, high peak and then leveling off?
That depends where you are. There are places that are constant producers, so we don't really understand the history effect in there all that well. And there are places that indeed, like the burst of activity kind of dies out eventually. The weird thing about working with Arctic soil, sometimes you feel like you're actually working in a locked up wetland. Some locations in there, especially coastal areas, they do not really function as an just a forest soil or like our grassland that we are more familiar with. They have their hydrology very tightly, just regulating what they do. Yeah. I think there's another.
Hi, great talk. I was curious with the functional stuff that you looked at from the metagenome analysis.
Yeah.
Have you compared that, like for say an NMDC, to other data sets, or seen if there's anything sort of unique about content in permafrost or what it might closely resemble?
Yeah, so we have not yet, because as I said, we will start putting the NG Arctic data in first. That will give us really a nice basis to to start comparing all the different things. But what NMDC provides actually just a basis to compare with the same pipeline. As you can imagine, most people like ourselves, we just do what just we think that's a good way of doing science, and reiterate over time. It's nice to have an infrastructure that just everything is done in the same way by just people who just provided the data to it, so that you just trust to that process. And so we are excited to do just exactly what you said, just to see how we are be able to just learn a bit more about these systems in comparison. Yeah. Okay, thank you very much.
All right. Well, thank you so much to our speakers. It was really great. And thank you so much for being perfectly on time. That was much appreciated. So now I'd like to introduce Leah Johnson, who manages our user research program.
Thanks, Emily. Hi, I'm Leah. I am a postdoc at Los Alamos National Lab, and so I do also work with the BFI SFA. But I'm very excited to work with the NMDC leading the user research efforts. And so today I'll give you a sense of how we incorporate user feedback into the NMDC product development by talking about how we did that for our new app, the NMDC Field Notes. And then Patrick will give you a demonstration of the Field Notes app. And so at the NMDC team retreat, the team was kind of brainstorming ways to help streamline data collection and management processes and really help facilitate this adherence to metadata standards, because we know it can be complex, especially if you've never really had any experience with them at all. And so one of the bottlenecks that we kind of identified is that there's this like at that data generation stage when you're out in the field, and we really wanted to transform the way that field researchers would be able to manage and collect their data on the go. And so at the team retreat, oh, sorry if I just laser pointed you. So we brainstormed, you can see the action here, what this app could look like and how we can facilitate this metadata collection out in the field. And so this was really designed as a spinoff of the NMDC submission portal and be able to integrate with that NMDC submission portal. And so something I've really enjoyed about this team is the breadth of expertise and diversity of backgrounds. And so combining that with our existing NMDC infrastructure, it really provides us this unique opportunity to build out this resource for the community. So to kind of get started, we wanted to gather community feedback and make sure that this app is actually going to meet the user's needs. And so we had this overarching question of how can NMDC Field Notes help support this digital metadata capture and reduce costs, labor, other resources for the field research community. And so first we performed a landscape analysis. Obviously, if there's an app that exists, we don't need to reinvent the wheel. But I wouldn't be talking to you if there were an app that already existed. And so a major thing we found is that our app will be free. A lot of other apps we found had, some sort of subscription cost or like kind of pay-to-play type setup, or other apps that we saw just really weren't meeting those needs of like helping to actually make metadata standards easier to use. And then we also collected some information on design aspects, because if we can incorporate flows and architecture that users are more familiar with already, it's a lot easier to just get up and running and use it right away. We also created some user interface mock-ups. So Patrick will show you a much nicer version. This is like a glorified PowerPoint presentation, basically. To help kind of facilitate discussions with our users and get feedback on our initial scoping. We also wanted to collect some information about current field sampling practices across diverse environments. So we wanted to know things like, what are your typical team sizes and roles? Like, who might need to access these studies? What are the sort of study types? Are they longitudinal? Like, will you need to manage them over a long period of time? We also wanted to know, like, how are users actually capturing their metadata in the field right now? And so we found that that's typically just, taking a table or a piece of paper out to the field or a notebook and just writing it down. And then you have to then digitize that later. And there's often also like a following QC step from another teammate. So it is this very complex process right now. And so we interviewed 12 different researchers across diverse roles, research questions, and environments, because we know that this is a very diverse field. And we wanted to make sure that we were capturing enough information to truly inform this app design. And so we had 5 researchers who were part of the Champions and Ambassadors Network, and then we had 7 from the broader research community, so including DOE SFAs and LTER sites. We also had a lot of diverse ecosystems. So we had researchers doing desert grassland research all the way up to sampling from lakes in the Arctic, which sounded very cold. And then all of our researchers had expertise across data management, field sampling, data processing, data deposition. And so they were able to give us really great insights kind of across the data generation lifecycle. And so from our discussions with these users, we gathered 50 insights to help guide 18 action items for development. And I don't really want to give any spoilers because Patrick is just about to talk to you about the app. But they really helped inform the app architecture and study organization and management and study export. Kind of more qualitatively, we also heard some metadata horror stories. So we had one user who told us that they lost their notebook with their field sampling key, and so their samples were really not workable anymore. Another user told us that their metadata sheet flew off their ship, and so they lost all of the metadata for the samples they'd collected so far. And then there was another user who had a water leak in a building. This is not funny. We laugh so we don't cry. Yeah, but a water leak in a building ruined all of their notebooks, ruined the computers and all of their backup drives, and so they just lost like a ton of history of work. And so really just speaking with these users, we found that we can really streamline that digitization process by, putting the digitization in your hand, in your phone, and then allowing easy syncing and exporting later. We can improve metadata accuracy in the field because it can be hard to know, especially if you're a first-time user of metadata standards, what exactly you need to collect, how you need to format them. From the metadata horror stories, we think this will help prevent data loss and support good data management practices, and also facilitate submission of your metadata to the NMDC data portal, as well as to those primary repositories. And so without further ado, I'm going to pass it off to Patrick Kalita, and he's going to give you a walkthrough of the current prototype. I don't know if you need that. Okay.
Should I try messing with this to see if we can? Hoo-hoo. This. Wow. Great. Thank you. Appreciate that. Yes. Hi, I'm Patrick. I'm a software developer for the NMDC project. So at this point, we're going to give you a little sneak peek of the in-progress development of the app. So here it is. Can everybody see this? Okay, great. Well, let's do this video thing instead. So. There we go. So this is kind of showing the ladder. This is kind of like our tips for working out in the field. And then heads up, to access the main content of the app, you would sign in with Orchid. So, and thank you for pointing that out. I did not notice that. Uh-oh. Hello? Don't know. Yeah, Julia, can you back up a little bit? Sorry, this is going getting way ahead of where I wanted to be. Where was it? Oh, so I'll just keep going. Hopefully they can hear. So So getting to the main content of the app, you sign in with your ORCID credentials. NMDC has kind of really embraced ORCID as an authentication provider. So if you've ever used the submission portal, you've seen that you can log in with your ORCID credentials there. So, okay, you can go ahead and play it. Speaking of the submission portal, so the app is designed to sort of work seamlessly with the submission portal. So once you've logged in, if there's, so I created some studies through the submission portal, and then you're seeing them here in the app, and you can, so you can see the study level details, you can see sample metadata, and if you drill into a specific sample, you can see the sample level fields there. Now I could add samples to one of these existing studies here if I wanted to, but I'll just walk through creating a new study through the app. Hit play, yep, so I hit new study. I can type in details, all that. And if you want to pause it here real quick, so then it'll ask you to select a template. And so these templates are sort of is a collection. Each of these templates is sort of a collection of metadata fields. They're all based on MixS standards and kind of customized for different environments. So this is kind of what we consider like best practices for sample metadata collection. And the templates, they also encode sort of validation rules to ensure good like metadata hygiene. I'll touch more on that later. And if you've used the NMDC submission portal before, this list of templates should look familiar because this is the same set of templates that the submission portal itself uses. So, okay, Julia, you can hit play. So I'll just go ahead and hit the soil template here. And then, yep, tap on save. Okay, so now I have my study, and I can go ahead and start adding samples to it. So I've created a new sample, and you can see with these orange icons that like already with my new sample, some like validation is happening. And so if I tap on, okay, can you play it, please? So if I tap on like sample name, okay, pause there, please. Now I have this like a little overlay where I can edit the value for the name, and this also like gives me a description of like what should be entered in this field, and also gives information about if there were any validation issues. Like in this case, this is a required field, so it's telling me that. Okay, I can go ahead and hit play again. So fill in a value there, and my validation message goes away. I can just go ahead and save that. And then as I'm scrolling through this list of metadata fields, you can see it's a pretty extensive list. Oh, go a little bit further, actually. Yeah, okay, pause it there. Where was I in my notes here? Oh yeah, so if I edit this growth facility field, this is a field that we expect to be one of a certain set of values. So this is gonna, okay, hit play again. So this is going to present those options to you. So you can only pick, the valid values. Other helpful features here. So if I edit the collection date, there's a button there to just say, oh, set this to today, because you're probably collecting the sample right now. And then when you go to edit like latitude and longitude, you can have it use your device's GPS features to just populate that for you. So that's really the kind of device-assisted like metadata collection that we want to continue building into the app. I'm sorry, actually keep playing it. I don't know why I said pause. So anyway, so fast forward a little bit here. Let's imagine that we've like added like 10 samples at this point. So you can see all the samples I've collected so far. And if I'm looking for a specific one, I can use this little search functionality to find that first one that I created. And then, if you need to, you can go back and edit the metadata for that sample. And if I, accidentally say, change the name to from 1 to 2, which I've already used 2. I'm getting some more validation here where it's telling me that you can't have like a duplicate name. So again, just trying to enforce good metadata hygiene, like right from the get-go out in the field. And let's see, what else is next here? Right. So, but of course also, as we know, mistakes do happen sometimes. So let's say I just totally messed up one of these samples and I just want to delete this. So I can just tap that delete sample button, get rid of it. Are you sure? Yes, I want to get rid of that. Okay, so that's kind of a brief walkthrough of the app itself. And I mentioned that this is all very much tied into the NMDC submission portal. So if I get back to my desk later, load up the submission portal, I can see the study that I just created here listed along with the other 2 that were there previously. And if I step through this interface, I can see the study level information that I edited through the app. And on this final screen, I can see my sample metadata that I entered through the app and the values that I set out in the field. So it's all just syncing sort of in real time as you're editing it through the app. And it is designed to be tolerant to offline use, because we heard from a lot of our potential users that you might be in a very remote location, you might not have cell service, so it is kind of designed to sync when it can, but it'll save it all locally and then kind of sync up later when you're back online. So we are taking that into account as we build it. So yeah, that's just a really, a very early preview. We know we have like a few more features we want to add in before there's any sort of public beta, but yeah, that's kind of the direction we're heading in. I think that's it. Oh, there we go. Okay.
Yeah, so just a brief thank you to all of our user research participants who provided a lot of really valuable feedback for us. And then also, we've just highlighted the NMDC Field Notes team members, especially our software developers, Patrick Klita, Ian Zhu, and Eric Kavana, who put a ton of work into getting this app even to this point that you saw today. Next slide. Where does this go to? Okay. And then we are expecting to have early access to the app in fall 2024. So the QR code just goes to that Field Notes webpage. But if you want to learn a little bit more and volunteer to beta test the app, definitely sign up at that link. So I'm going to give it back to Emily to moderate the discussion period. But thank you.
Okay, so we have plenty of time for questions, thoughts, input. Okay, right off the bat, great.
Yeah, that was great. So when you were showing us the sort of tutorial there, you're selecting, when you were showing, that was great, awesome app, looks really cool. When you were showing us the tutorial there, we're selecting like what type of sample this is and where it's from. How much of that can be just skipped and maybe filled in later when you're back at the lab, so you're not fumbling around in the field trying to fill it in? Just wondering about that.
Oh, dual microphones.
Look at this. Hi.
Yeah, no, that's totally. So that's and sorry, thank you for asking that question, because that's I wanted to make that point a little more clearly. But that's exactly the idea. Is that through the app, you collect what's relevant to collect in the field, and then once you're back at in the office or whatever, you can use the submission portal to fill in.
So it'll let you
everything
jump
else.
around without necessarily filling in everything right then.
Absolutely.
Okay.
And some of the things that we're thinking about, what hasn't been implemented yet is ways to sort of like cut down that list of metadata fields, so you're only displaying the ones that are, you know, kind of relevant for field collection. That's one thing. And then also, you know, the ability to like copy certain fields from one sample to another, like if they're going to be the same for every single sample. So that's work that we already have like planned for this.
Okay, great, thanks.
So I was wondering if any thoughts wasn't there to help users a bit more in the accessibility, in the sense that like, is there a plan or thinking or discussion was made to like fill these, you know, just fields with like just voice commands? Because I can tell you as a person who needs to hold a lot of material from one place to the other, hands are very valuable tools in sampling, and you don't really necessarily want to keep them too busy with, you know.
Yeah,
Yeah.
no, absolutely. And that was something that came up when we, you know, in our sort of potential user interviews, was this, what we sort of like started referring to internally as muddy gloves. So like people who just have like, you know, mud all over their hands, but they're also trying to use a device. So as far as, you know, specifically solving that with like voice recognition, we talked about it. I don't know. We might look into it, but like it's not like specifically on our list of things right now, but it's kind of, it's percolating in the back of our minds.
Hi, great app. It looks really awesome. One thing I know seems like the core of the app is based around the template and the standardization, making sure everything's filled. As someone who's worked out in the field and seeing like the ability to use this on a cell phone, a lot of times we take photos or notes too of like, for instance, if a plant's supposed to be alive, but it's dead, or it's something's moved, or we dug too deep or something. Is there some ability to kind of take, you know, maybe that's not a part of the template, but sample-based notes or add photos as well?
Yeah, it's also something we kind of have talked about, and that's a little more in the concrete plan column than than like voice dictation. But yeah, definitely attaching photos to at the study or sample level, and maybe just like a sort of freeform notes area, which always makes like the like metadata and standardization people cringe a little bit. But you know, that will probably happen. Yeah.
So I'm curious, when you were talking with the users, and you mentioned that there are a bunch of like apps that exist, and I'm just wondering if when you had those conversations, if people had tried using applications like the ones that you found in the field, if you talked to anyone who had experience with that, and what you might have learned from, you know, sort of real world experience with an app in the field.
Yeah, so none of our users actually do use an app in the field because they haven't actually found anything that fits their needs. So that's kind of what we wanted to do, is to actually design something that would meet what they're actually, you know, writing down in the field. And so like a lot of people will take out either a notebook, or they'll have like a pre-printed template of, you know, they're like, oh, we know that we always need to collect like location information or whatever. And they just write that down. But then when they get back to the lab, they then have to transcribe that, like, so take it from their little sheet and then put it into like a Google Sheets or an Excel file. And so really the app is basically going to replace that little template that they take out into the field.
So yeah, but so maybe like the scope of like having met their needs, I think, is an interesting one to explore from kind of like a usability perspective. Like, and I don't know that you all went into this with the users, but you know, those evaluation criteria when looking at an app, or what were they thinking when they looked at it and saw it and they were like, oh, this won't work for me, right? And this idea that just came up of, you know, taking a picture or taking other notes, like, did you guys get the chance to explore with the users, like, hey,
What if we just asked you all to send us a picture and then we extract metadata from the picture first, just to kind of like get an understanding, like do they have phones in the field and what that looks like? So anyway, I'm always curious about user research, so I'm sorry to like drill down on this. I'm just kind of wondering like what else you could have gleaned about the existing process and sort of how they make decisions, and then how you present this app, right, so that they make that decision to download it and use it.
Yeah, that's a great point. I mean, because Aaron had just mentioned, like he takes a lot of field photos. A lot of the other groups we talk to, they will also take photos. So they all have their phones already available, and tablets. People will have, some people will even have like their computers. Yeah, and so it is something that would kind of integrate in with those existing practices, but just kind of save time. As you can imagine, like you can put your metadata into this app, you can take the picture, so it's all kind of centralized, and then.
Sorry. I think one of the things that we consider to be like the value add over existing apps or just using like the Notes app on your phone is the the metadata templates, and making sure that people presenting these things that people like might not think to collect in the field, but when they're presented with it in in the app, they might think about it.
But I like filled out the metadata templates, and it takes a while too. So anyway, I guess I'm just like super curious, and I want to just want to see people use it while they're doing an experiment. So I feel like maybe that's like the next step, right? Of like find some volunteers amongst the champions and go out there and just watch them, which I know sounds kind of creepy and weird, but it's like,
Yeah.
what are you doing?
I think
yeah.
Anyway,
sorry,
I think in
stop.
broad strokes, yeah. I mean, our plan is to do beta testing with like ambassadors and/or champions, and get their feedback. Yeah, see what happens, and we'll take that feedback when we get it.
So we have two questions in the chat online that we'll answer really quickly. So one of the questions was, does data in the NMDC need to be JGI generated? The answer is no. So you can work with us. We do pull in a lot from the user facilities. So we work really closely with the user facilities and are trying to streamline that process as best as possible. But we do have data from outside of the user facilities, and we'll continue to do so. And then we'll also continue to pull in JGI and EMSL data. Anything else for that?
Jessica, I'll just add to that if you are a JGI or EMSL user, that this is compatible with all of the information that both JGI and EMSL would need in order to submit samples for either a ficus or a CSP or an LSR project. So we've been working very closely. So ideally, if you, summer sampling season would like to, test the app, it would be great, and we'd be happy to, get in touch with folks to be able to do that. But for future, a lot of this is future studies that would be generating data, and ideally hitting that sweet spot with, we see a lot of latitude, longitude errors. Wouldn't it be great to just be able to click that, get the latitude, longitude coordinates right on site, and that could feed directly into the submission portal and JGI and EMSL for future projects.
And then we did have another question in the chat about taking in data from remote sensors, so things, pieces of equipment that are taking data in the field, and how that would work with the app. So Patrick, I don't know if you want to take that one.
Again, it's another thing that was brought up in our user research, and yeah, thinking about it, but at this point, I don't have a great answer for that. There's a wide variety of devices and sensors out there, so
I just want to echo that. I mean, I never been almost to the field, but I'm doing being a data scientist. I want to echo that the lat long is very important to get right, and I saw all kinds of things happening there, and it's important because it's one thing you can link to things like remotely sensed data that's out there potentially. So if there are resources for just one thing, I'll get that right.
Yeah, real quick. Have you looked at like battery usage since we're out in the field and outlets might be limited? Just wondering.
I don't remember battery usage coming up specifically, but that is a good thought. Mm-hmm. Yeah. Yeah. Maybe we should put put it. Yeah, it should continue to work in airplane mode, and we'll test that, before we release anything. But maybe we should put in our checklist for going out in the field. Bring a bring a portable battery bank with you.
Hi, very interesting app, and I'm field scientist, so we work a lot in Siberia or Svalbard, Canada, and some of these sites doesn't have even internet connection. Will your app work like without internet connection in a very remote area when we can, and I like Nathan, question about voice recognition, because this is important. If you drill soil, usually this is like teamwork, a few people drilling, another subsampling, and this is very important. And maybe if there is some option, just talk to phone in internet without any internet, and then when you're on the base, you can connect to internet and improve your data and spreadsheet and everything.
Yeah, absolutely. So the way it's designed is that when you first log into the app, you would need to have a connection, and from there it should fetch everything it needs. And then if you're out in the field and you're disconnected from any service, it's designed to just store all that locally until you are reconnected to a network, and then it'll sync up at that point. So it's designed to work that way, and obviously we're still in early development, and so it'll need rigorous testing around that. But that's definitely something that we designed in from the ground up. And yeah, I hear you about the voice dictation thing, and so we've heard that a number of times. So I think that's something we should probably look into.
Okay, I think we will call that a wrap. So thank you everyone, both in person and online, for joining. Thanks for all your great comments. If this is something you're very interested in for the Field Notes app, please reach out. We are actively recruiting, people who have requests who would like to use this within their research. So we are very open. This is, as you heard, in active development, and we do have expected early access in fall of 2024. So thanks again to Josué, Buck, and Neslihan for really great talks. And thanks again. We have recorded this session. We do plan on posting again for those who had issues getting online at first. I really apologize, but we will make this full town hall available very soon. So thank you again. Please help me thank our speakers and our team with another round of applause.